Right. Welcome, everyone. My name is Abhishek. Tom has already introduced. Thanks, Tom, for the introduction. So I'm a PhD research candidate with Professor Robert Henry, based at Coffee Institute at University of Queensland. So my research is primarily focused on to study the evolution of genome size diversity within the Proteaceae family. So where I've been working on more than 50% of the generic diversity in the Proteaceae family, which is found in Australia. And we're just trying to look into the genome size diversity and the potential significance of that diversity within the same family. So today's talk is more about the how do we measure the genome size in plants using the flow cytometry, which is a technique which widely used in the genome size estimation. Acknowledgement part. I would like to acknowledge traditional landowners, and I pay respect to their ancestors and their descendants. So we begin by the quantifying the genome size. What is actually a genome size, and how do we denote this? What are the units for this? So in this picture, we have seen like whatever the entire DNA content within a nucleus is, we call that as a genome size. Let's say if a nucleus, a haploid nucleus, has got 4 chromosomes in it, and whatever the DNA content of that nucleus would be, that we will call it as a 1C value. On the right side, when we have a diploid cell, the number of sets increases, which is double in this case, and that DNA content will become 2C value. In terms of measuring this in terms of units, then we measure them into picograms. 1 picogram, sorry, there is a typo here. It's 1 picogram equals to 978 megabase pairs. Now, considering the diversity, this image has been taken from one of the research. We found that there is a profound diversity in the plant itself. So this image just represents the plants themselves, where we found like nearly 2,400 fold diversity in land plants in terms of their genome size. Some of the genomes are really small, as 60 megabase pairs, whereas some of the plants are really large, 150,000 megabase pairs. What are the significance of the genome size? It's usually associated with the cell size, stomatal size. So if a genome size, if a species has a larger genome size, then the cell size would be larger. And that can have some implication in terms of physiology and morphology. And also the growth rate, when we have a higher DNA content, when the cells have higher DNA content, they need to like take more time in terms of reproduction or synthesis, DNA synthesis. And it can also have the terms of evolutionary impact, because excessive DNA amount can give genomes that leverage where they can try on new genes, neogenesis, what we call. So it has, in terms of application, it has some implications for breeding. Let's say when a genome is large, a small genome is comparatively easy to edit, whereas like some of the really large genomes must be harder to edit, or in terms of breeding purposes. And it also enhances the sequencing cost, because larger genomes are more costly to sequence, and it also improves the complexity, because a lot of DNA content in these large genomes is made up of repeat contents, which is kind of hard for sequencing as well as assembling. Now, there are some methods how we can measure the genome size. One of technology method we will be discussing is flow cytometry, but there are a few others as well: spectrophotometry, whole genome sequencing, and k-mer-based analysis. K-mer-based is like where we just sequence either whole genome or some part of the genome. So now we're just moving on to flow cytometry. What is it? And how does it work? So it's a fluorescence intensity-based method. So conventionally, what we do here, we just take a fresh leaf material and we chop it fine in a suitable buffer that contains a lot of detergents and other components. So that helps to release nuclei, intact nuclei from this fresh leaf material. Once we get that filtrate, we pour that into cytometry tubes and we add a dye or staining agent. Like usually for the DNA content estimation, we are using propydium iodide. Then we load this to flow cytometer. So flow cytometer works usually on a principle that we have to excite this fluorophore, the propydium iodide. So at a certain wavelength, we can excite the propydium iodide, and it will emit some kind of light. So that detector in the flow cytometry will detect what is the property of that light in terms of their scattered, like either the light scattered in forward direction or in the side direction, and the intensity of the light. So based on the intensity, we see. So usually when we are chopping the leaf material, we're chopping two things at the same time. So we are chopping, one is a test species, for which species we just need to know the genome size. And the second species is the internal standard, for which we know everything like, let's say, genome size and the ploidy. So we need to know these two informations about that internal standards. We chop them together. So in that suspension here, we have the nuclei from both species and we stain them and then we load them to flow cytometer. And then based on that, we see some different histograms here. So we can see in this image, we can see two peaks, so P1 and P2. However, this first peak is a lot about debris. So where we have a ruptured nuclei or other cell components or secondary metabolites that also can bind with the propydium iodide. And then their fluorescence is usually lower than the intact nuclei itself. So here in this case, we have P1, it's our internal standard, and then Nippon bar rice, and we know the genome size of this species. And so how do we calculate the P2, the genome size of test species? Just by getting the fluorescence ratio between P2 and P1, which is roughly 2 times. So then the genome size would be roughly 2 times of the internal standard here. So it sounds a very fairly simple process, but the issue is with the plant cells, the recalcitrants, the secondary metabolites, where we have the issue. Because a lot of the times buffer are not compatible enough, or sometimes our plants are having secondary metabolites that could really hard make it challenging. So we are not able to detect any peaks because a lot of the because of the excessive debris, we are not able to detect any peaks in this case. So when we have a limited sample material, if we, let's say, if we have collected limited leaf material from the fields, then it becomes really challenging to identify, let's say, in terms of herbarium samples, which are really always harder because we don't have that leverage of having excessive material. And also for the high throughput study where we're just processing a lot of species altogether. In this case, having a challenging species could take us longer time to do the genome size analysis. And the third thing is lack of logistics and processing facilities. So usually we collect our samples from the field conditions where we do not have enough necessities or facilities, so we can bring our leaf material back to the lab in fresh condition. Let's say if we take it more than 4 or 5 days and we are not able to preserve the leaf material, then the nuclear integrity will can be impacted significantly. And in case if we don't have intact nuclei, then flow cytometry doesn't give us the results that we seek. So what are the limitations of conventional approach? As I said, they're sensitive to leaf recalcitrants, secondary metabolites, and such as tannins, and these can degrade the nuclear integrity and also can hinder the capability of propydium iodide to bind with the DNA. Sometimes we just need optimizations for high throughput FCM. Let's say because I was working on more than 40 species, and these species have different morphology and different leaf chemistry. So one buffer does not work with everything. So in that case, we just need to seek some optimizations for better results. And also reference standards are one thing. So because in the conventional method, so since this method is like has been widely used for 20, 30 years, we do not have enough reference standards. And even if we have, then in that case, we do not have the verified genome size. That's the one, because inaccurate estimates of the standards can lead us to inaccurate estimate of our test species. So as a part of the PhD, I worked on a method where we have used frozen material. Let's say if we are not able to preserve a nuclear integrity of the leaf material, then we have a method like we can preserve a leaf material usually by freezing the leaf material in liquid nitrogen and then keeping it into -80 conditions. So in that case, we can maintain the integrity of the nuclei. And we using the frozen material, we isolate the nuclei and we process them for the flow cytometry. This method has a preprint on bioarchive. Uh, the only issue with this method is when we have leaf material in frozen condition, then we have a higher debris amount. And then this happens. So in this image, right? So in this image, we can see that there are two peaks. However, at the bottom of the peaks, we have excessive debris. That's the issue. We're not able to clearly identify our two peaks. And when these peaks base are wider, then the coefficient of variance goes well beyond 5%, which is not a standard in flow cytometry measurements. It is usually recommended that the CV percentage is below 5% for reliable results. So how do we deal with this debris thing? Because this is a conventional analysis. So we opted for a automated peak modeling and debris compensation that is done by a software called Flowploid. It's an R package. So here the software finds itself, like it just identifies the peak itself based on considering that if we just provided the peaks of the top of the peak, the top point of the peak, it will consider them as a normal distribution. So it will think, okay, this will be a normal distribution, and based on that, it will just give us a clear idea of where the peak is, and then it removes all the debris part from the peak. And in this, using this method, we can still maintain our CV percentage below 5%. So that's how we can complement this frozen method with a specific analysis here. Another useful advantage like of the frozen material is we can process much larger like amounts of much larger volumes of the leaf material with this method, because we are homogenizing grinding material. However, in the previous instances conventional approach, we were using certain, let's say 40 milligrams of leaf material or 60 milligram of leaf material, and we were chopping that manually. And each time when we chop the leaf material, we could have a difference in terms of chopping itself, and we may have different chemistry in this nuclear suspension. It gives us a little bit different results from whatever we have done in earlier, let's say one or two, three replication. Each time we'll see some sort of difference in the genome estimates. However, in this case, uh, we process all these replicates in one process. So I have taken nearly 2 grams of leaf material, and then we can make 5 replicates in one go. So using this method, we can increase the precision of the genome size estimation. However, we found this was not a significant difference, but it still increased the precision. Yes, as I said, like, so we have a larger volumes of tissue to work with. That's why we can release thousands of intact nuclei. When we have higher nuclei per peak, then we have a leverage that we can uh, squeeze our peaks a little bit to reduce the CV percentage well below 5%. We can make multiple replicates in one process. High debris content, but that's the disadvantage of this technique, because freezing can damage the cells. So we see high debris and the lower resolution of the peaks. But we can sort that with the debris compensation analysis. And there's one thing, we didn't try that because our method was based on where we just freeze the leaves and the next day we process that. But in some studies, it's been found that if we have stored leaf for longer time, let's say 6 months or longer than that, then the nuclear integrity could further degrade. So this is something that we did not look into, but it could be looked into in further studies. Logistically, it's challenging because if we are in the field, then we need to have access to liquid nitrogen and dry ice and -80 conditions. Because once we freeze the leaf material, we cannot let this thaw. If it thaw, then the leaf material is of no use at all. But having said that, it's a very big advantage in terms of sequencing studies, because the same leaf material can be used for flow cytometry and the same leaf material can be used for genome sequencing purposes, because at freezing leaf material, we can store or preserve the DNA and RNA structures. So this method is particularly useful for breeders, those who are working in the fields and they cannot maintain the leaf material integrity for longer time, let's say more than 4 or 5 days. So they have a solution that freeze the leaf material and bring it back to the lab and process it. And then same nuclei, isolated nuclei can be subjected to the DNA isolation protocols further. So based on this method and the conventional approach, I processed nearly 43 species, representing 43 genera from the Proteaceae family. So I've just given 3 examples here. I haven't put the whole list here. So in our previous study, we found out the genome size for Adenanthus, which was found the smallest genome in the Proteaceae family, was 0.59 picograms. Uh, However, we used that was against Pisum sativum. So internal standard was P, the toast tag P. We took Nippon bar rice in our case, given that it has multiple genome assemblies. So our estimate was nearly 25% smaller than the previous estimate, and the same for Hollandia. And one of the critically endangered species, Eidothea, that I have sequenced, and de novo genome assembly for this species has been done. We found out the genome estimate for this species were nearly 36% overestimated. So then it was a question for us, like why there is a big difference in the genome estimates from previous uh, estimates. So there are things, usually it happens in flow cytometry. Sometimes we take different cultivars of internal standard. Let's say instead of Torsteg P, we could have taken some other cultivar, and that cultivar could have a different genome size. Sometimes it's all about technical errors, um, or maybe we have incorrected calibration of internal standard, because if we have initial value incorrect, then the test species will also be given incorrect values. So we found out, like in 20 years or late 90s, there was a lot of work done for the internal standards of flow cytometry. Uh, and the previous calibration for P was against human male leukocytes. At that time, human genome was considered as 7 picogram. But the current values of human male genome, where we have like whole genome has been sequenced and it's gapless genome with the use of long read and advanced sequencing technologies. So now it's been considered to a 6.15 picogram, which is way smaller than the previous estimated. So all the previous standards that were calibrated against a human male leukocyte, we believe that were incorrectly recalibrated. So that's why we took another approach to calibrate a few more internal standards for plant genome size estimation. This is a C data. So I'll just give a little bit background about this. So this is C database from Kew Botanical Gardens, and this database contains the values, genome size values for plant kingdom. and there are like nearly 12,000 genome size estimates for plants, but barely any of them is based on whole genome sequencing approach. And the reason is obvious because in past the sequencing technologies were not that superior in terms of uh, capturing the highly complex region of the genomes. Especially in the recent advancements of long-read sequencing technologies such as Oxford Nanopore, PacBio, HiFi reads HiC, we are able to sequence complete genomes and really some of the really complex genome parts, example, centromeres or telomeres, and we can also effectively do the phased assemblies. So that was the whole idea of this paper. So where we took the Nippon bar rice genome, so Nippon bar rice has been sequenced uh, frequently, 3-4 times at least. So the first instance was based on short read sequencing, which was done in 2005. At that time, the rice genome was considered as 0.795 picograms. And one of the recently complete genome was based on uh, long read sequencing technologies, but that was based on consensus sequence. So how the consensus sequence work, we'll talk a little bit later. And there was another paper from the lab, Robert Henry's lab from Abdullah, um, which is based on haplotypes. How do we like haplotypes of the rice, Nippon bar rice. So all these three approaches were giving us different genome estimates, and uh, and that's the thing. So in terms of genome size, when we calculate estimate genome size from genome sequencing, we need to have account for both of the haplotypes, because both of the haplotypes in one particular genome could be different from each other, as Abdullah found. Whereas in one of the earlier study in 23, despite the genome was complete, but that was based on consensus. So consensus genome could unintentionally favor this longer haplotype. So it works in way like, when we have a longer haplotype, and it can avoid the smaller haplotype. So that's a reason it always account for the longer. It gives a favorability to the longer haplotypes, and it gives us incorrect value of the genome estimate, which is not right. So we have used these three values of Nippon bar rice, and we calibrated 5 different internal standards here: Arabidopsis thaliana, and then sorghum bicolors, particular cultivars BT623, which is sequence, Gossypium cotton, and P, the Torsay cultivar, which was used in 2015 study for Proteaceae genome size estimation, and then we have Nicotiana as well. Uh, and then we found out that these 5 standards were overestimated significantly, and we found particularly Pisum sativum, the Torsay cultivar, which was previously thought to be around 9.49 picogram, and our estimated recalibrated estimates say that it's 7.22 picogram, which is 31% smaller than the previous value. So that's the reason we found out why we see the big discrepancies in our first study, where we were just taking the whole species of multiple species of Proteaceae family. Right. I would like to, I think so. That's the thing, that's the message I wanted to provide through this slide. And I would like to acknowledge my supervisory panel and collaborators, and the support got from Botanical Gardens, University of Tasmania, and Tropical Herbarium. And I would like to acknowledge the UQ facilities and all Robert Henry's lab members. Thank you very much. And any questions?